Okay, so when seeing this title, you may wonder what is the link between climate and fish skeleton. Indeed, fish growth is almost continuous during their whole life, but it is also strongly driven by the seasonal fluctuation of the environmental conditions. So the fluctuating growth rate of fish is reflected within their own bones through the alternation of different growth marks. And for instance, in tropical area, there are the largest growth marks in blue that are laid when the conditions are favorable, so when the growth is fast during the wet season. And on the contrary, during the dry season, the growth slows or even stops, thus laying a thin line, sorry. So we could use this fluctuating growth rate recorded in fossil fish bones to describe the evolution of the intensity of seasons in the past. And this would constitute a new way to infer paleoclimates in continental context. But first, this new approach needs to be calibrated on extant species that are closely related to the studied fossil taxa. And for this purpose, we chose to study the Nile perch because it is widespread in the Nilo-Sudanese area and because it dominates the fossil fish assemblages since the Miocene. So we tested the consistency of the growth rate within one individual, within its bone and between the different types of bones, and also the correlation between the growth rate and the climatic condition it's under. So the study specimen was captured in 2014 near N'Djamena in Chad. And we sampled different bones that are often found in the fossil record and that allow the identification. So here I present you the preliminary results I have for the vertebra. I have been able to identify the last 2 to 3 growth marks in the vertebra. And given the estimated age for this specimen between 3 and 7 years old, this means that each main growth mark may have been laid each year. And this is consistent with the seasonal regime in N'Djamena with 1 wet and 1 dry season per year. And we can see that the growth rate in 2012, so this growth mark, is less important than that in 2013. And this is not expected because in fish, the growth tend to decrease along their life. And but we can see that in 2012, in N'Djamena, the temperature dropped and the rains were less abundant this year. So this may have resulted in a stress for the animal and thus a less important growth this year. So to conclude, this study showed that it was very difficult to identify and to interpret the growth marks for this specimen. So we will need more specimen fish captured at different months during the year to better understand when exactly in the year are laid the main growth marks before being able to use this future paleoclimatic proxy on fossil fish bones. And thank you for your listening.
Okay, thank you again to the organizer for this opportunity for me to talk at this conference, which is great. Despite I specialize in seasonality by means of cementochronology and microwave analysis. Today I will not talk to you about this project. But I will switch to another topic, which is a preliminary results of the study on the work diaphysis founded in the Nadale Cave, which is a Middle Paleolithic deposit in the north of Italy. Okay, as you can see, the Nadale Cave is a single-layered Kinami-Syrian site dated to 70,000 years ago, that is, say, MIS4, which is peculiar because it's one of the rare sites from the MIS4 in the north of Italy, and it is located in a region already rich in Paleolithic evidence, both from Middle and Upper Paleolithic. During the excavation, we unearthed like 30,000 bone fragments, and Neanderthal deciduous tooth, and some lithic implements from recognized as a Kinami-Syrian technology. Moreover, I studied the seasonality and the extent of the site occupation, and 335 bone retouches. Today, I'm going to present to you the bone industry, since it's a study that is at its very beginning, and I would like you to share any ideas, any suggestion or comments that you may have about these materials, that is quite peculiar and rare for the Middle Paleolithic, as you all know. I have really few comparison at Grenal, I guess, and at Chajinskaya Cave in Siberia. We founded 77 worked shaft that bear some anthropic modification. All these shafts belonging to limb bones from large size herbivores. That is, say, giant deer, red deer, and bovids, which are the main taxa in the deposit. And we were able to distinguish between intermediate tools, flaked bones, and blunt pieces. For now, the study is really preliminary, so I just separate this three category. And I started to measure the angle, the mapping angles, to, I don't know, to infer some recurrency. We will see going on with the study. As I was saying, the main taxa are ungulate. 46 diaphysi were not determined, and the others belong to red deer, giant deer, and bovids. So for the future, I would like to try some experimental activities to better understand how the tools were made and used and gripped, because some of these retouch that these fragments have on the edges seems not to be functional. So my hypothesis for now is that the retouch can be to better grip, to better handle the tool, and also is sometimes this kind of retouch, non-functional retouch, is associated with the retoucher stigmata. So this is an hypothesis that I would like to evaluate during experimental activities. And then I would like to apply some use-wear analysis to see if there is some residue or something that I can tell on the functional edges. So thank you for your attention. I would like you to share your opinion about this, and if you have any comment, any suggestion, are welcome. Thank you.
So good morning, everyone. I'm Jessica, and this morning I present you my master thesis project, so an ontogenetic perspective on the emergence of sexual dimorphism in the modern human cranium. So a little introduction. So the cranium is one of the most used and most important skeletal structure in sex determination. but only few studies have been focused on the emergence of sexual dimorphism in a human cranium from an ontogenetic perspective. So this project, our project, aimed to analyze the differences in cranial morphology between sexes, so male and females, from 8 to 20 years old. For what concerns our sample, so our sample is composed by a total of 50 individuals. We use 3D models of the cranium from 50 total body CT scan from recently deceased individuals. We have 25 males and 25 females by the New Mexico Descendant Image Database, that is a very important online open access database for anthropological studies. And we use geometric morphometric approaches and some statistical analysis to try to investigate sexual dimorphism between sexes. So for each cranium, we define 50 fixed landmark and 1,000 pair surface semi-landmarks that you can see in the image. So with the yellow color, the landmarks, and with the pink and green color, the semi-landmarks. And we perform a GPA and a PCA, that are the generalized procastis analysis and the principal component analysis, to investigate the relation between the cranial morphology and the two variables, sex and age. So for what concerns statistical analysis and our results, we see that age and size represent the 16 and the 26% of the total variance. Meanwhile, the variance associated to sex is the 7.4%. So, and this variance is explained by the intersection of the sex with size and age. So, sex, age, and size are related together. In this plot, you can see the PCA, so the principal component analysis in form space, and you can see males and females, so the males with red color and the females with a black color, and the different ages of the different individuals. Along PC1, we have size and age. Meanwhile, along PC2, so PC2 represents sexual dimorphism. And what we can see is that the multivariate trajectories between males and females are not statistically different. Another important result, so the evaluation of the morphological differences between males and females during growth reveal as highest differences at 9, 16, and 19 years old. So in this graph, we can see that we have 3 different peaks in which males and females are different each other. One important thing is that we have to underline that for the 9 years old, we have only 2 individuals, so 1 male and 1 female. Meanwhile, for the other age ranges, we have 4 individuals, so 2 males and 2 females. And this, at the end, could affect the results of the analysis probably. So the results in general suggest us that the differences in the ontogenetic growth between sexes differ in size and also in shape. And what we can see in this video is the surface warping variation. So if you focus on the video, you can see that the cranial changes during growth in female and male. And especially, you can see the different areas in which the surface is changing. So with the red, yellow, and orange color indicate the local expansion of the cranium. Meanwhile, the purple and blue color indicate the expansion, the local expansion. So to conclude, in the future, our results need to be confirmed. So including more individuals, using a bigger sample, and also extending the age range of the ontogenic trajectory, including individuals from 4 to 7 years old. And an important thing that I want to underline in the conclusion is the advantage in the use of open repository like a New Mexico database, that is very important, and also the use of free software like R or MeshLab for the application and development of new research projects in archaeology. This is all. Thank you very much for the attention. Thanks to the organizers and my tutors, and happy Women's Day. Thank you.
So thank you and welcome to Widening the Perceptions of Coloring Materials. so building a use-where analysis reference collection. So the experiments were undertaken to understand the relationships between ferruginous raw material and the surface of different abrasive tools. The creation of wear marks by abrasion was observed to document characteristic marks resulting from this particular processing method. These experiments were additionally designed to test the reliability of interpretations of use-where analysis on coloring material. This was used to build a reference assemblage, all the while controlling fundamental parameters that I will go over next. For the experimenter, though, the continuous goal was to produce a fine and regular powder. So 4 coloring materials of different petrology were chosen for the experiments. The reference collection served to test the relevance of resorting to quantitative analysis to help with the descriptions of formed striations. It also will be used to supplement the experimental sample collection of the pigmentothèque to compare the petrophysical properties of different coloring materials in the geological reference collection. So the effects of prehension were also tested during these experiments by creating 1 or 3 facets, the movements, the timing with intervals large enough to have a difference in powder production, the addition of an adjuvant, so either of animal fat or of water, and also testing the granulometry of abrasive tools by artificial means such as sanding paper or natural such as sandstone. So macro and mesoscopic observations were undertaken to identify and define characteristic marks from abrasion. An analytical framework was put in place to characterize the use-where traces to better comprehend the way that coloring material surfaces imprint. Each material showed specific reactions to the abrasion task, but each of them showed a characteristic barcode that forms from the removal of material by the abrasive tool. It became apparent that the interactions between the abrasive tool and the worked material is complex, and that their intrinsic characteristics must be understood before being able to interpret the use-where data. With this qualitative analysis, it was possible to distinguish abrasion marks coming from different abrasive tools, from different movements, and from different work times. But it was not possible to distinguish the abrasion marks from the use of different adjuvants or to distinguish the formation order of the multiple facets. When it came to measuring the striations, 10 of them were measured from edge to edge on each sample. For the data from the adjuvant experiments, a Kruskal-Wallis test was performed and ascertained that there was a significant difference between the ferruginous sandstone and the other materials. This led to the conclusion that the nature of the raw material had an effect on the width of the striations, more so than the addition of an adjuvant. For the samples abraded on sanding paper, a Kruskal-Wallis test was also performed and verified that the facets abraded on the coarsest paper had significantly larger striations than the ones abraded on medium or fine paper. This demonstrated that the size of abrasive grains did in fact have an effect on the width of the striations. With this quantitative data, it was not always possible to identify precisely the granulometry of the abrasive tool from the striation width, mainly because the coloring material also plays a role in the abrasion process, leaving striations from its own grains. So to answer the question, are we able to distinguish between different abrasive surfaces? The experiment showed that this was indeed possible, but only for certain parameters, as mentioned previously. It is therefore imperative to have a qualitative repository that is precise and exhaustive to fill the lack of information and the biases brought on by purely quantitative observations. The qualitative approach makes it possible to account for the properties of the materials worked, their behavior during abrasions, as well as to identify the main trends. Future experiments will focus on repeatability in order to have an extensive quantitative benchmark model to answer specific hypotheses, making the science all the more reliable and relevant. Thank you very much.